All right, good morning, everyone. So today we have the privilege of your classmates giving a presentation regarding platinum-based anti-cancer strategies. And as part of this discussion, we will hear some insight into overcoming resistant mechanisms. And so what I'm going to do is I'm going to turn it over to your classmates. They will actually present. I'm just showing the PowerPoint, but I'll have Rafael and Kiara take over. And then that'll be followed by Kisha and Miguel, and then finally by Andrea and Jael. All right, then. So I'm going to stop projecting, and then either Rafael or Kiara can go ahead and present. All right, then who will be presenting?
Rafael will be presenting the presentation in a moment.
Okay.
Sorry.
Can you see the presentation?
Yes, it's not in slideshow mode, but I do see it.
Okay, now?
Yes, I see it now. So as soon as you start, I'll let you know when you have 2 minutes remaining, okay?
Okay.
Rafa, can you put it again in full screen?
Oh, yeah.
Thank you. Okay, well, good morning, everyone. As the professor said, we are going to be presenting the seminar on Platinum 2 and Platinum 4 based strategies for anti-cancer treatment and overcoming resistant mechanisms against metal-based anti-cancer compounds. There will be 3 themes presenting today: Rafael and myself, Kiara, Kicha, and Miguel, and lastly, Andrea and Jael. The articles that will be presented today, their titles are the first one, which will be presented by us, is titled Emerging Platinum for Prodrugs to Combat Cisplatin Resistance from Isolated Cancer Cells to Tumor and Microenvironments. The second article, which is Kisha's and Miguel's, is titled Platinum 2 Thionate Complexes, Synthesis, Structural Characterization, and In Vitro Biological Assessment as Potent Anti-Cancer Agents. And this article is about the complexes that were synthesized and later treated by human cancer cell lines. They will talk to you about how they show promising anti-tumor effects in comparison to cisplatin. Later, Andrea and Jael will present the novel Platinum 4 prodrug with self-assembly property and structure transformable character against triple-negative breast cancer cells. And this article is about the novel conjugate cisplatin adjuvant-based prodrug that have the capabilities to self-assembly in nano-sized structures, which is very promising in overcoming some problems that are faced with cisplatin. As you can see, all the articles that will be presented today have something to do with cisplatin, which we are familiar with because of the class. So now we will start with our article.
Okay, so good morning, everyone, again. Here we are going to present this article. As Chiara said, this was the article that the professor sent by email. We are quoting this paper today, so everything that we're going to talk today is about this paper in specific. So we are going to start with the cisplatin and drug resistance, the introduction. As it said, the anti-tumor activity of cisplatin was discovered in the 1960s. It was discovered, cisplatin and its analogs that are carboplatin and oxaliplatin, and these have been used as anti-cancer agents in the clinic. So about a half of the chemotherapy schedules includes a platinum-based cancer drug. Because of the great success of cisplatin, researchers started to study more the mechanism of action and to develop more potent platinum-based agents to overcome the issue of platinum resistance. But how does cisplatin enter the cells? Well, cisplatin enters cells by both passive diffusion and active transport with the help of copper transporter CTR1, as we discussed in one of the lectures, and then loses its two chlorides. We're going to see that in a moment, and binds purines in the genomic DNA. This binding of platinum to DNA results in a structural distortion of the double-stranded structure, leading the inhibition of the DNA replication and transcription, and the subsequent DNA damage response that leads to apoptosis. It is suggested that cisplatin resistance derives from multifactorial cellular alterations that includes reduced cellular accumulation of platinums, detoxification of platinum by TIL-containing molecules, and elevated levels of DNA damage repair. We're going to see the picture in a moment, but these factors all result in fewer platinum-induced DNA lesions in the genomic DNA, leading to cancer cell survival. In many cases, more than one mechanism is involved, and additional factors such as changes in chromatin structure, DNA hypermethylation, and histone modification. Here, as we saw in the paper, in the picture at the left, there is the mechanism in which the cisplatin loses the two chlorides, as you see in the middle, and in the right side you have the resistant mechanism, and we're not going to discuss because of the time, but almost all of this has been discussed in the lectures by the professor. And in the second point, platinum-4 complexes to improve cytotoxicity and to overcome resistance. Platinum-4 anti-cancer complexes are prodrugs of platinum-4 derivatives. We're going to discuss what prodrugs are. Platinum-4 prodrugs have a central platinum atom in the relatively inner number plus 4 oxidation state, and in the cell, the prodrugs are activated by cellular reductants to generate the active platinum-4 species. And after cellular entrance, platinum-4 complexes are subsequently activated by biological reducing agents such as ascorbic acid, cysteine, glutathione, and other macromolecules. The octahedral platinum-4 complexes are inherently more resistant to ligand substitution than square planar, as you see there, which limits the premature reaction of platinum with biomolecules before arriving at the cellular target and minimizes the undesired side effects. The 2-extra axial of platinum-4 helps to fine-tune the properties of the compound, including lipophilicity, redox potential, target specificity, and fluorescence.
Now, the prodrugs which Rafael mentioned are composed of the platinum-4 and targeting groups. These complexes that are formed have a specific ligand that show improvement in cytotoxicity in comparison with cisplatin. It's important to know that these biomolecular interactions with the receptors that are overexpressed in the surface of the cancer cells, and that because of this, they are able to increase the cancer cell selectivity of cisplatin and help overcome the low cellular accumulation to combat the issue of resistance. In essence, this conjugation of the ligand and the platinum 4, they result in a prodrug which has the ability to enter the cancer cell specifically through receptor-mediated pathway, which doesn't happen with cisplatin, and this is why the prodrugs are effective. On the next slide, we have some examples of these prodrugs, and these are the biotin conjugated cisplatin-based prodrugs, and the first one is Platinum Bio 1, which showed a 3.3-fold improvement in toxicity against breast cancer cells when compared to cisplatin, but it has an inconvenience, and it is that the in vivo anti-cancer potency of these prodrugs is very much unknown because the complex of the tumor environment, it may have an effect, and it may compromise the effectiveness of these prodrugs. Now, those prodrugs can be divided in dual action or multi-action prodrugs. And in this image, we can see 6 prodrugs, 3 of which are dual action and 3 that are multi-action. A, B, and C structures are dual action, and D, E, F are multi-action prodrugs. I will now focus on dual action prodrugs, and we should remember that these complexes have ligands, and those ligands are bioactive. They are in the actual position, and because of them, they are able to overcome drug resistance. They show enhanced cytotoxicity when compared to cisplatin, and because of this, it is difficult to determine how much the bioactive ligand contributes to the cytotoxicity of the conjugate. They are trying to optimize the ratio of the platinum and the bioactive ligand in order to determine how much it contributes to the cytotoxicity. One of the dual action prodrugs is chalcoplatin. This contains 2 p53 activators in the axial position, as we can see here, and this is very beneficial because p53 is actually the one in charge of the cellular response to the cisplatin-induced DNA damage. This prodrug presented a 31.6-fold higher toxicity in cells in comparison to cisplatin, and it also showed a low resistance factor. We also have another prodrug called monochalcoplatin, and this one is able to enter the cell via active transport. It's actually very similar to the first one, with the difference that it only has one chalcone group. It presented a 422-fold higher toxicity compared to cisplatin in human cancer cells, which is even more effective than the chalcoplatin. And this remarkable cytotoxicity is due to the high level of DNA damage that it can cause. It also has a very low resistance factor in comparison to cisplatin. And the third dual action prodrug, which we will talk about, is the nucleotide excision repair inhibition platinum 4 complex, and this is able to enter the cell efficiently and release both the cisplatin and the nucleotide excision repair inhibition into the cell. This prodrug presented a 88-fold improvement cytotoxicity in cisplatin. non-small cell lung cancer cells, which have a higher nucleotide excision repair capacity. It also shows a lower resistance factor in comparisons to cisplatin alone. And this project is very effective because the way it works is pretty much cisplatin causes the damage, and the nucleotide excision repair inhibition, it stops the repair that the cisplatin caused, which is one of the reasons that there is a resistance to cisplatin. Now we're going to present a graph that we created to compare cisplatin with the prodrugs, and we can see that the resistance factor of cisplatin is very high in each of the cases, and the platinum complexes are much lower, which helps us understand that the overcoming of cisplatin resistance is indeed achieved with these complexes.
Okay, so a prodrug can be defined as a drug substance that is inactive in the intended pharmacological actions and must be to convert into the pharmacologically active agent by metabolic or physicochemical transformation. So now talking about the multi-action platinum-4 prodrugs, as Chiara said, to improve the cytotoxicity of and overcome resistance, novel platinum-4 complexes with multi-action properties have been constructed to hit several therapeutic targets simultaneously. These multi-action prodrugs of platinum-4 complexes exert several biological activities at once to achieve different functions. For example, alteration of cellular glucose metabolism and cytotoxicity and level of platinum DNA binding. Combine multiple effects, including depletion of mitochondrial membrane potential and activation of caspase, killed the cancer cells, as we know. So these effects validate the multi-action property of this drug. Two of the studies that were made, in the first one, the resistant factor for the conjugate in the A2780 cis-R cells is as low as 0.86, and the value for cisplatin was 4.9, suggesting the potency of this strategy against cisplatin resistance. In the other one, with the SGC7901 CDP cells, the resistant factor was equal to 7.37, and for the cisplatin was for the prodrug, was 0.29, indicating its ability to overcome cisplatin resistance. So finally, the cisplatin resistance derives from multifactorial cellular alterations, therefore simultaneously targeting different resistant factors. These multi-action platinum-4 prodrugs present significantly increased cytotoxicity and promising potential applications.
In the article, they talk about the delivery of platinum-4 prodrugs by nanocarriers, but we're not going to focus a lot on that. But we did want to mention that the nanocarriers are able to enter the cell through endocytotic pathways and deliver the platinum directly into the cisplatin-resistant cells, which is very efficient because the reduced platinum accumulation in cancer cells is actually one of the key causes of cisplatin resistance. And the nanocarriers that are between 50 to 200 nanometers tend to accumulate in the tumor tissue. These delivery nanocarriers are able to improve the cytotoxicity and overcome resistance. And this is due to overcome resistance in the monolayer cell models, but their in vivo anti-cancer efficiency remains unclear because the complexity of the tumor environment is still unknown how it affects the prodrug. the tumor microenvironment, we have an idea of what it is, but we did want to mention that the mechanism of the resistance is well studied, but the main problem is the tumor microenvironment, because how it affects the ability of the drug to interact is unknown, because the microenvironment has a different acidity and oxygen concentration than the normal tissues. And many non-cancer cells contribute to the tumor growth. Now we're going to talk about the platinum 4 complexes and how they help activate the immune response. And as we know, the tumor cells are able to evade the immune surveillance, and this is why they keep growing so much. But they do mention that immunochemotherapeutic platinum 4 prodrugs of cisplatin have been reported and show promise in treating cisplatin-resistant tumors. And this is very promising because it can result in an efficient treatment for tumors.
Okay, so now talking about platinum complexes targeting hypoxia. The hypoxia frequently occurs in solid tumors due to the lack of blood supplies to the growing tumor nodules. The conventional chemotherapy that is commonly used may be less effective against hypoxic tumors, and cisplatin-induced apoptosis is believed to be strongly dependent on the growth rate. Platinum-4 to target hypoxia is a very promising strategy to overcome resistance to cisplatin, especially under hypoxic condition. And finally, platinum-based drugs have been evidenced to directly stimulate the immune cells to activate the immune response and improve therapeutic effect. Platinum-4 target complexes can be activated in the tumor microenvironment outside of cancer cells to target those factors and activate the immune cells, are also promising to conquer drug resistance. Finally, with the conclusion and perspectives, cisplatin has been used in the clinic for more than 40 years. This is not new. And however, the non-targeting nature and acquired inherent resistance limit the clinical performance of cisplatin. The more inherent octahedral, as I said, platinum-4 prodrugs represent a promising way to solve drawbacks of cisplatin, as they can be modified with a range of ligands for the purpose of targeting cancer cells, alteration of non-targeting nature, and acquire inherent resistance in that. Early studies of dual-action platinum-4, as Chiara said, show improved cytotoxicity, especially in the cisplatinum-resistant cells, and the conjugated bioactive ligands exerted the expected mode of action inside cells. In the other side, the multi-action prodrugs were developed by conjugating multiple bioactive ligands to achieve specific cancer treatment and overcome resistance. As a conclusion, as we said, drug resistance of tumor cells is driven by multiple factors. Therefore, direct conjugation of co-delivery of platinum-4 and multi-action platinum-4 prodrugs, this is a promising strategy to improve cytotoxicity and combat resistance in this field. Thank you.
All right, thank you, Chiara and Rafael. So we're going to move on to our next presentation. That will be offered by Keisha and Miguel. So who's going to be presenting?
Yeah, I'll be presenting right now.
Yeah.
Okay, can you see?
Yes.
Okay, well, I'm gonna introduce us. So, my name is Miguel Nieves, and well, my partner is Kisha Fernandez. We will be presenting the article, The Transplatinum DNA Complexes, Synthesis, Structural Characterization, and In Vitro Biological Assessment as Potent Anti-Cancer Agents.
Okay, in what regards to the method-based anti-tumor drugs, as said earlier by Chiara and Rafael, is cisplatin. That is currently the best drug and the most studied drug. However, it has some major limitations. So, to explain this better, it is well known that the biological effectiveness of the complexes of platinum derives from the formation of the drug and DNA cross-link adjuncts. They interact with DNA bases, the guanine and adenine, as mentioned by the previous group, and that generates DNA lesion and subsequently cell death. In what regards to the activity and toxicity of the platinum drugs, they are directly attributed to the nature of the ligands around the platinum center. In consequence, this is why it was introduced in this study the transconfigured platinum 2 complexes, because they have displayed considerable in vitro and anti-proliferative effects against a wide range of cancer cells. So the unique cytotoxicity profile of transplatinum 2 complexes with bulky planar ligands are attributed to their different structural and DNA binding properties in comparison with cisplatin analog. You can go to the next slide.
Okay, so to get an idea of kind of how the synthetic scheme is, we have a figure here from the article. We at the research group first synthesized the complex labeled as the A by ligand exchange, and they did it by a reaction with the diphenyl group with a known complex of platinum, and they used dimethyl sulfide and dimethyl sulfoxide to displace the monandate phosphate ligand. After they synthesized this group, they put it in these conditions, which is a ethanol solution with potassium thionate salts, which produces a complex of general trans at the platinum complexes. These are all the platinum complexes done. They are all pyridine or at the or biotenosyl groups, and mostly we will be focusing on the 1A group, since we will explain later why this one will be the most seen.
In general, in this study, these platinum complexes shown here in the middle of the presentation, in the middle of the slide, were synthesized and were characterized by NMR and X-ray crystallography. These complexes were treated by human cancer cell lines A549, SKOV3, and MCS7, and shown the promising anti-tumor effect in comparison with cisplatin. This is one of the advantages that these trans complexes have over cisplatin. So in this starting complex A was synthesized through ligand exchange. Treatment of this complex in dichloride with ethanolic solution of potassium cyanide salts produce these complexes, these subsequent complexes, labeled as 1A, 1B, 1C through 1E. And in what regards to the single crystal structure determination, this complex A, crystallize in the monoclinic, while the complex 1A through C crystallize in the triclinic crystal system. You can go to the next slide.
Right, so for other experimental sections they analyzed by computational details. They did many different biological assays, and they determined the intracellular reactive oxygen species of the platinum complexes. What they did with the computational details, they used a program called the Gaussian, and they performed density functional calculation using B3LYP theory, level theory. And by using this, they were able to fully optimize the geometries of the complex. They were also able to optimize this by using different analytical derivations, and they also took into effects such as solvents and other things. And for the biological assays, I won't try to get too into details, but they did a number of them. And to start off, they did have to purchase 3 different cancer cells, which came from breast cancer, ovary cancer, and lung cancers. And oh, my bad. The first one they did was they determined the cytotoxic activities of transplant complex in a 96 wall microplate, and they used it to measure the absorbance of the individual walls at 490 nanometers, and they measured the IC50s of each compound for each cell. Another biological assay they did was the apoptosis assay, where they used the apoptosis detection kit, and they mostly tested it out with the 1A compound, as I mentioned before, in a well kosher plate, and they treated it with different conditions of the 1A and also used an untreated sample as a negative control. They also did another cell cycle analysis where they measured the MCF7 cells, and they also used untreated cells as a negative control, and they stained the DNA cells and not RNA cells, so they could kind of get an idea of how they impact the DNA instead of RNA. They also did a shift mobility assay where they used cisplatin as the positive control and untreated samples as the negative control, and they also measured the 1A. They also did another assay, which was the COMET assay, where they tested out the genotoxic effect of the compounds, and this time they used another compound which is called doxorubicin as the negative and positive controls. And finally, as I mentioned, they did the intracellular reactive oxygen species. They used an assay kit where they determined the oxidative stress response of SCOV3 cell lines after treating it with 1A, the 1A compound, and they again used the untreated cells as the negative controls.
Okay. To support the cis trans isomerization of these platinum complexes, as mentioned earlier, they use the density functional theory, the DFT theory, by the optimization of the lowest energy structures, either in gas phase or dichloromethane solution, of the proposed cis and trans geometries for, but specifically for the complex 1C. In this case, we will be only studying the complex 1C. This DFT calculation indicated that the trans isomer is remarkably more stable than the suggested cis isomer. Additionally, the NMR spectra confirmed this calculation. So this is one of the other advantage that we were talking about, that these trans complexes have over the cis platinum complexes. It provides the better thermodynamic stability. And Mika, you can go on. Okay. In this case, this is an R-type representation of the complexes, which is an oak-rich thermal ellipsoid plot. This representation is the molecular structure determined by X-ray diffraction. In this case, the geometry around the platinum center in the complexes is considered slightly distorted square planar. As the despite by perspective or the view of the structure, the allele substituent of the PPH2 allele phosphine ligand, as we can see, are detected to be perpendicular to the molecular plane in each structure. You can go to the next slide. Okay, to study these compounds and these drugs, they have to study the cytotoxicity to see if these compounds in fact is useful or not. In this case, the in vitro cytotoxicity activity of these compounds were evaluated on 3 cell lines. The cell lines mentioned earlier. In this case, the complex 1A is the one who show higher anti-proliferative activity than cisplatin on the study cell line. It show a good anti-proliferative activity with an IC50 less than for the measure for cisplatin compound. The IC50 is a measure that indicates how much of a particular inhibitory substance, in this case the platinum compounds, is needed to inhibit in vitro a given biological process. In this case, the cell death by 50%. These indicate that these new compounds have a lower IC because it means that you need less amount of the compound to have the desired effect on the biological system. So this is another advantage that these new trans platinum compounds offer against the cis and platinum compounds. Next slide, please. In this figure, this is shown a flow cytometric analysis of apoptotic effects of this complex 1A on MCF7 cell line. These results show that with the increase of the concentration of these compounds from concentration from 2.5 to 10 micromolar, the percentage of cell deaths in early apoptotic phase significantly increased in the treated cell. This observation indicates that this compound, as a representative of the new transplatinum 2 series, is able to effectively induce apoptosis in cancerous cells in a dose-dependent manner. This is one of the other advantages that these compounds offer. This is more offers more apoptotic effects. You can go on to the next slide.
So another one of the biological assays they did was the cell cycle analysis. And this is a method that is commonly used to assess the anti-proliferative effects of anti-cancer drugs. So what they did is that they use a fluorescent DNA binding dye to stain the DNA and measure the amount of DNA present in the cell. So we should expect that the G2 phase should absorb twice as much as the G1 phase because the DNA content does double during that phase. And because the S phase is when it is splitting, the G1 cells should be less and should be more than the G1 cells, but less than a G2 cells. But when we compare the untreated cells and the 1A cells at both concentrations, we really don't see any different change in this. So we can definitely, they observe that 1A does not really have a clear effect on the cell cycle of cancerous cells. The next biological assay they did was the COMET assay. And this assay is used for the genotoxic effect of the synthesized compounds. And to kind of have an idea of how this works, a damaged DNA should have a longer tail, or comet, as they say, and a longer tail does display more genotoxicity, and untreated cells just seem as unfragmented cells. And we can see in this figure that the untreated cells, they're just pretty much unfragmented cells, while the other controls and the 1A and 1A treated ones just show a long tail or comet. In the case of the 50 micromolar, we can see that there is no of the cell nucleus present, which does show that this compound does target the genome content of the cells. The next biological assay they did was the electrophoresis mobility shift, and they did this to kind of see the direct interaction of the 1A compounds with DNA in comparison to the cisplatin. And once we observe this, we can definitely see the difference between cisplatin and the 1A compounds, and which this really clearly kind of shows that the synthesized 1A compounds do have a direct interaction of DNA and probably other molecules as well. And this is consistent with the other genotoxic effects. And they also did a kind of different study that was with the binding mode and binding site of the transplant compound synthesized, and it does show that all these synthesized compounds do have high interaction with this cancerous cells. So this assay does confirm it. And for the final biological activity they did was the reactive oxygen species, and they tested the SCOV3 cell line with the 1A compound. So what they did was they used a non-fluorescent dye, and they oxidized the ROS with peroxide, and this kind of shows that the 1A does induce ROS moderately, but however, higher concentrations of 1A don't necessarily work better in comparison to lower concentrations. So they did determine that the lower concentrations of 1A do in fact work better than higher concentrations, but
So, in conclusion, a synthetic approach has been introduced to obtain these new platinum complexes in the transconfirmation. They were characterized by multinuclear NMR spectroscopy. In addition to prove the antiproliferative activity of these compounds, they were tested against three human cancer cell lines, the cell lines mentioned earlier. And in fact, it showed potent anti-tumor properties. Specifically, the compound 1A was the one who has the most anti-proliferative activity. And in general, the evidence showed that these new compounds in transconfiguration provides more advantages over cis platinum compounds, and it provides better properties of anti-tumor. In the next slide, you will see the reference.
Okay, well, thank you so much, Keisha Miguel. We're going to transition to the next presenter. So next we have Andre and Jael.
Yeah, I'll be presenting.
Okay, so hello, good afternoon. My name is Andrea Mazer, and myself and my classmate Jair Rodriguez will be presenting from the paper of the novel Platinum 4 prodrug with self-assembly property and structure transformable character against triple negative breast cancer. As my peers have mentioned, cisplatin is a therapeutic that's commonly used to treat cancers such as ovarian, lung, breast cancer, among many others. The research group had shown it was particularly good at treating triple negative breast cancer, or TNBC for short. It is characterized by coming up negative for progesterone and estrogen receptors, as well as the growth promoting protein, HER2. Other breast cancers that are positive to these tend to respond well to hormone therapy. But in the case of triple negative breast cancer, despite that it's only 10 to 15% of the cancers, it's considered the most aggressive since it has the highest chance of spreading and a high recurrence rating, making it essentially the worst breast cancer you could be diagnosed with. The way that cisplatin is able to treat this disease is by entering cells by passive and or protein-mediated transport, such as those concerning the copper transport receptor 1, as previously mentioned by our peers. After it is hydrolyzed, it enters the nucleus and cross-links DNA, altering the structure of the DNA, which can lead to damage once the enzymes that are trying to repair it end up cleaving it, which can not only lead to apoptosis, but also reduce the proliferative characteristic that is very much characteristic with triple negative breast cancer. Despite all of its great uses, it suffers from many undesired side effects, mainly due to low specificity, which has caused people to lose their hearing, have to receive dialysis in order to mediate these symptoms, and not to mention the common nausea, fatigue, pain, irritation. And not only that, we also see resistance from these types of cancers. Next slide, please. Thank you. So cells can become resistant to cisplatin by a variety of methods, as previously mentioned by some of our peers. Some of them include the downregulation of the production of the copper transport protein, copper transport receptor 1, more excretion of the drugs, and enhanced DNA damage repair. And so because of these varied capacities of resisting the drug, multiple research groups have developed, next slide, please. Multiple research groups have looked into using other strategies to reduce this resistance. Some approaches, other than developing other drugs, have been to modify cisplatin itself. In other cases, they've looked into using more bulky ligands. They've looked into combining it with other medications, using them both at the same time for an amplified effect, and the use of nanomedicine. Next slide, please. This group has looked into the use of the non-hormonal male contraceptive adjuvant, or ADD for short. This compound is a substrate of mitochondrial hexokinase 2, which serves an important role in glucose metabolism and can initiate the first step in the glycolytic pathway. Why is this important? It can result in reduced ATP production and an increased reactive oxygen species level within cells, which is what this research group found. Next slide, please. Next slide, please. Oh, no, never mind. Oh, sorry, there was a lag. ADD is a hydrophobic drug, whereas cisplatin is a hydrophilic drug. And so by conjugating these, the product was a amphiphilic compound, which means that it has both hydrophobic and hydrophilic characteristics. And it's particularly important since it gives this compound the ability to self-assemble, and thus using nanomedicine, you're able to deliver this compound to the cancer cells and treat the cancer.
Okay, so as a form of introduction, I want to change gears a little bit, and we're going to talk a little bit about nanotechnology and drug delivery agents. So since the professor has basically alluded to what drug delivery agents are, I'm just going to give a brief introduction. So drug delivery agents or nanotechnology are basically a very relatively new field in which we have seen that drugs are becoming less efficacious in their ability to treat diseases because of many different reasons. Part of it may be resistance, lack of drug absorption, or even just not specific enough and causing too many off-target effects. So drug delivery is just a way that some researchers have found that they can overcome these limitations in drug treatment or disease treatment. Many of the drug delivery agents have many properties that are very sought after, some of which are systematic delivery of drug supply. Sometimes you don't want to be giving a patient many different shots about a specific a therapeutic agent. You might just want to give a drug delivery capsule that may have all the drug they'll ever need, and then release it at a certain rate that you can control. Also, reduction in the amount of drug needed for a patient, because if you have increased drug absorption due to the nature of the drug delivery particle, then you can have better stability of the drug and better absorption. Also, and very importantly, reduce off-target effects. So you can modify these particles most of the time using synthetic methods or even inorganic methods, depending on the particle, to be able to specifically target an area or a target that you're trying to treat. And of course, some even have increased drug potency depending on the particular application that you're trying to use that drug for. It may be cytotoxicity or actual remediation of a target. So I'm going to talk a little bit about what types of nanoparticle delivery systems are. This is very brief. There are different, a nanoparticle can be composed of different materials. So these materials can be lipids, can be polymers, inorganic materials like metals or carbon nanotubes, and even small molecules like that can form dendrimers. And even some of them have been used as some proteins have been used as drug delivery methods. So there is basically an infinite array of combinations that you can use to have different types of nanoparticles for your specific application. Usually they're in about the 100 nanometer to 1 nanometer size, but some of them vary in sizes and can be larger. A very important aspect is shape, because shape will, the morphological properties of a particle will give it certain characteristics that may be more llamativo for a specific application. And a really I find really cool part about material science and nanoparticle science is surface modifications. So even if you have all these different types of combinations, you can even further them by adding different groups on the surface that may recognize certain specific targets. They can have functional groups that may attach to certain proteins, pegylations that may increase stability, etc. So now that we have covered nanoparticles as a general, there's a certain sector of nanoscience that's known as self-assembly. This relates more to a supramolecular chemistry, which is a very obscure side of this scientific discipline. So it basically has to do with the discipline that works with molecular assemblies. So it's a supramolecular property where a single molecule or a monomer can self-recognize and form higher ordered structures. And these are usually driven by non-covalent interactions, known as hydrogen bonding, pi-pi stacking, or hydrophobic interactions. And these can be initiated through various different ways. So you can have even more versatility in the method that you're trying to employ. It can be redox, a change in the solvent, like is the case in our paper, having metal ions that can mediate the formation of these assemblies, temperature, or even light. So it is very versatile, and it can offer you various methods for its implication in drug delivery. So now, really briefly, I want to cover what is the main idea of this article. So the authors have synthesized a small library of platinum-4 prodrugs using platinum or cisplatin and adjuvant, which is this molecule right here. And they have basically synthesized different compounds which have the same structure but only vary in these in the specific chain length of the aliphatic chain length. So they can vary by having 2 carbons, which is the ethyl, 4, 6, 8, or even 12. It's important to note that the C4 and the C6 are very important for this article because these have the properties that I was talking about, the self-assembly properties. They have the non-covalent interaction properties that are specific, that they give secondary structures. Think of like proteins that can give you beta sheets, or they can give you alpha helixes due to those non-covalent interactions of the hydrogen bonding. Well, the same or analogously happens here. You have basically a structure that has the properties to form secondary structures. And they also talk about this dimer right here, but I'm not going to go into much depth because they don't cover it much in the paper. So think of this molecule as this little representation here that they made for us. So the carbonyl length, the chain length can vary. So when you have aggregation or self-assembly under different conditions like pi stacking hydrophobic interactions, they can form these like the little molecules are like all stacked on top of each other, and then they form these nanoparticles. And as I was talking about earlier, you can modify the surface of these particles using different methods. They're using pegylation, because pegylation gives you stability, it gives you more hydrophilicity, and can overall increase efficiency of the drug that you're trying to carry inside the target. So once you have this nanoparticle formed with pegylation, you basically have the drug inside, and then you can go on to delivery. So for the C4 and the C6, they have what's known as a structure transform, which I'll be covering in a later slide, in which when you have this structure transform, you can even induce further apoptosis. And as my partner already covered, what is the mechanism of cisplatin? It will cause DNA damage through that to that cross-linking. But since you also have adjuvant, the adjuvant will go to the mitochondria, as we have already alluded to, in past anti-cancer metal drugs. You can have increased reactive oxygen species that can, cause mitochondrial dysfunction, lower the mitochondria potential, which that can lead to release of cytochrome C, cause cell apoptosis. So this is basically what these particles look like. So you have the CN, or this can vary at different lengths. So for the C2, C8, C12, and the dimer, they all form these spherical little particles that are like really cute. They form, so they stay like this no matter how long time passes. They still keep their spherical shape. But for the C4 and the C6, when giving enough time, or if you give them, an increase in temperature or increase in concentration of the original monomer, you have these secondary structures, these wires or these sheets. And this is very important because secondary structures have been previously shown to increase cytotoxicity through different mechanisms, as increase apoptosis. So even though you have a dual action of having the adjuvant, which causes reaction oxygen species and causes DNA damage, and cisplatin, which also causes DNA damage, you can have secondary structures which give you even more efficacy in the treatment of cancer. So this is some of the experiments they did. So in the in vitro copper transport receptor, so they wanted to see if they used 3 different cell lines in these experiments. First is the A549, which is basically just a small non-small cancer lung cancer cells, hepatic carcinoma cells, and MDA-MB231 are basically cisplatin resistant triple negative breast cancer cells. So as you can see, CTR1 is relatively not expressed as much as the other 2 proteins, and this is basically why they chose this particular cell line, because it doesn't doesn't respond as well to cisplatin, and this can be seen in some further experiment or further data that they show. And but they also saw, which I didn't quite, I'm still trying to wrap my head around this, is that they showed that for all the three cell lines, they all show the different, the same or non-significant platinums or cisplatin concentration in the cells. So they did some other experiments where they saw the gamma H2AX, which is basically like a biomarker of DNA damage, and they saw that as exposure of cisplatin in 8 hours, you have increased DNA damage, and in 24 hours you have really a lot of DNA damage. But for the cisplatin resistant cells, this is all for cisplatin. It is not as apparent or as marked. So they did another experiment where they saw, what is the platinum concentration with different the different the different particles or nanoparticles that they synthesized in this paper, and they saw that for all the particles there was increased platinum concentrations inside the cells. So this means that this delivery method is working. We're delivering more cisplatin into the cells, and we're seeing it right now. So another experiment they did is that they wanted to see, what does this look like in cells? So they did some transition electron microscope experiments, and for the C2, which as I alluded earlier, they're still spherical. They still have these little spheres that you can see all around them. This is still the TNBC cisplatin resistant cells. For the C4, you still have these still secondary structures. So this is like the wire structure that we saw earlier. And for the C6, the plate or the sheet-like structure. And what they saw is that for the C4 and the C6 in this confocal microscopy experiment, the green fluorescence is coumarin 6 labeled nanoparticles. So the nanoparticles, they labeled them with coumarin, and they saw that as time increased, the concentration of the particles increased in contrast to the C2, which increased a little bit but not as much. So the C4 and the C6 are basically what is going to be the main focus of this article due to that structure transform. So they also want to study apoptosis and the mechanism that conduces apoptosis, and they saw that.
Yeah.
Yeah. Hello?
You have 2 minutes remaining, Jael.
Okay. So they saw that, this is a flow cytometry experiment. So they saw that basically that the C4 and the C6 had increased apoptosis or apoptotic capability compared to all the other, even cisplatin or ADD or the control. And they saw that as well. We had for the nanoparticles, we have this is basically an experiment of reactive oxygen species. So we have also an increase in reactive oxygen species. And lastly, we have a decrease in mitochondrial potential, which relates to the rupture of this mitochondrial membrane. And this side, we have that the C2, which is basically the particle, had increased Cleaves caspase 3, which is the main form of caspase that does apoptosis or biomarker for apoptosis. They did some in vivo tumor growth inhibition studies where they saw, how the tumor volume changed in mice, and they saw that in PBS or cisplatin ADD, that change was relatively, insignificant. The tumor kept growing, but in the platinum nanoparticles, they saw that the C4 and the C6 specifically, the tumor decreased in volume significantly, while still remaining the same body weight. So the mice were still healthy. As a concluding remarks, basically the research group synthesized a small library of platinum-4 products that self-assemble into nanoparticles, showing increased cytotoxicity and effectivity in killing triple-negative breast cancer cells. And these drugs are really cool because they showcase dual action by causing apoptosis through ROS production DNA damage, while also a structure transform, which increases even more the potency. And I think that as an opinion, these innovative techniques that are at the crossroads of scientific disciplines. You're taking things from inorganic, bioinorganic, organic, and supermolecular chemistry are really, the path forward into more advanced treatments for different diseases. So thank you all for listening, and yes, that's the end of the presentation.
All right, well, thank you very much. So we're going to open this up for questions. Any classmate questions? All right, so I will start. So first of all, just a few pointers. You guys should use slide numbers because it's very hard to follow and to be able to go back and say a particular slide. So I've had to write down some titles. So that's going to make it a little bit challenging when I want to refer to something. Okay, so I want to ask Rafael and Chiara. So you mentioned P53 as something that's responsible for some of the resistance against cisplatin. But in general, what does that protein do, P53?
I'm sorry. Yes. P53 is actually linked with repairing the damage that the cisplatin can cause. It's called the guardian angel, if I am not mistaken.
Right. So my question is not having to do with cisplatin. What does P53 do in general?
Well, it's a protein that pretty much maintains the DNA structure.
Okay. Thank you. So it's part of the general repair machinery of the cell. All right. Another question that I have is, with these platinum 4 compounds, so I'm still with you, Rafael and Chiara. So Rafael, if you can answer this one. These platinum 4 compounds, are there any indications of similar biomolecular binding that can interfere with the speciation of platinum 2? For instance, any indication of albumin being able to bind the platinum 4 compound or the platinum 4 ion?
Can you repeat the question, please? It's my internet.
Right. So my question has to do with any structural speciation characterization of the platinum 4 compounds. As we've talked about platinum 2 speciation and how it can be quite complex, especially with regards to biomolecular interactions. Anything that you read in that article, but also to contextualize the work, that shows perhaps that platinum 4 as a compound or as an ion can interact with albumin.
I really don't remember reading something related, but with albumin. But it said that the complex with platinum is better than platinum like ion, like individually.
All right. So yeah, that's something that I will suggest you guys to look into a little bit more, right? One of the things that I do emphasize is the relative stability of the compounds, but also their ability to ligand exchange, right? Their kinetics with regards to that. Just to see with platinum 4, both platinum 4 and platinum 2 are in theory ligand exchange inert, but we have seen that even with that issue, there is ligand dissociation that affects how the metal might actually be uptaken into cells. So that was my question had to do with that issue. All right, Keisha and Miguel. So I'm actually going to give some constructive criticism here. You guys did a presentation about a series of compounds, which at most of the time I had no idea what these compounds look like. So that's problematic because it's very important that you show the structure of your compounds and really emphasize the ligands. That's something that I do in the lectures. I do show you figures so that you can get a general sense of structure. So that's something that was kind of really difficult with your presentation, as I had no idea what made 1A, 1B, 1C, and so forth different. So my first question has to do with what what exactly was different about these compounds, and what do you think was the reason why, let's say, 1A may have been slightly more cytotoxic or antiproliferative against some of those cell lines?
Okay, the scheme that Miguel present in the introduction,
Mm-hmm.
that scheme was the structure of the complexes. The complexes 1A, C, B through E,
Mm-hmm.
well, the only thing that changed was the ligand.
Right. And what were
That is
those
different.
ligands that were different?
The difference of this ligand was the substituent. In the case of 1A, the ligand is abbreviated as PY. The Give me a second to look for the name. Is very long name.
Right, right. I'm not asking for names. I'm asking for the ligand properties, because I understand that there was one slide that showed something about it, but you never actually discussed what were the distinctive properties of these ligands for us to be able to appreciate these. As far as that boils down to what is known as a structure activity relationship study. And for us to be able to appreciate further slides, we really needed to get some insight as to what exactly were these differences. So I know that there was a moment where you showed these compounds, but after a while, we have not actually internalized the material as you guys have. That's why you guys are doing the presentation. We need to be able to understand what are what specifically are we focusing on, so that as you move on, we can get an idea of what these results may correlate to as far as structure. So that's the insight that I'm looking for. And so I'm not asking for a name. I actually just wanted to know the general differences about the substituents.
Oh, okay. I think I understand the question. The difference of this ligand is the structure and the bulky. One ligand is more bulky than other. For example, the ligand SPY that I was talking about is substituted with the F3C. That is the substituent that it have additional to the in the ligand. The ligand general structure is like a benzene ring, but with a nitrogen. The structure shown in the scheme that Miguel presented. That structure of the complex 1A is the most simple one. So that is the structure that is not substituted by the ligand, and that is why it is shown in the study that it interacts better with the DNA, and thus, in consequence, that's why it have the better properties.
Okay, so Miguel, can you give me some insight? Because you did talk about some computational studies that were demonstrating some cis-trans isomerization. Why was that important? How does that have any impact on the bioactivity of these compounds?
Well, it didn't really you mentioned the computational analysis.
Well, you guys talked about it.
Yeah, well, it didn't that wasn't really used to measure like the biological. It was more to get a sense of like the geometries as to understand if it's like cis trans, but it doesn't really mention, well, at least from what I saw, it didn't really mention anything related to the biological activity. It was more on the structural information that was based.
Okay, and so, well then, give me some insight then about these trans compounds.
What do you mean?
Well, why are we looking at these trans compounds, right, in terms of activity, given that what I focus on in the course have been, just the basics of cisplatin being uh, more active than its trans isomer? Well, in this context, you guys are talking about these trans compounds, right? And so, to an extent, the isomerization that they will be referring to could have an impact on whether or not these compounds have a certain activity. So I think that's one of the fundamental reasons why they even looked at whether or not there is this isomerization possible, as it can have an impact on the compounds. But I want to know why these particular trans compounds seem to be active.
Hmm. I'm really trying to think here, but I, like I just know that mostly when it comes to the compounds, it was more of the folks, but I think it does change in structure and tracks better with the DNA, if I'm not wrong. Like, I think it interacts better with the DNA since of the double helix. I think that would be the most, like, my reasoning.
Okay. All right. So, Andrea and Jael. All right. So, with your presentation, I curious to know you mentioned that the toxicity that was, I guess, to measure toxicity, they were looking at the general mass or the general weight of these mice. That they did not change, therefore suggest that, generally speaking, the compounds were not toxic. Were there any additional, let's say, histochemical studies done to assess toxicity?
Yeah, they did another study with C2, but they didn't do anything with the C4 and the C6, which was my main focus of the article. But
Yeah.
they did do I forgot the name.
Histochemical analyses?
Histochemical analysis of different in vivo models of mice, and they
Uh-huh.
saw that in general there's not too much cytotoxicity for the overall health of the mice, but there is increased cytotoxicity for the cancer cells, at least in summary. But they didn't really go into much insight onto those specific. I mean, the original data of the C4 and the C6. They basically just said that they looked at body weight, but they didn't even mention. I'm basically just alluding to maybe I think that body weight is an indication of how well the mice is doing when giving the medication.
Right. So that's like a physiological factor that's typically measured, and then when you go more deeply, so if based on what you're saying, right, that there seem to be more effect on the tumor tissue as opposed to, like, let's say, some of the other organs, other tissue. What does that suggest to you then?
It suggests that basically there's not. I mean, in my opinion, that there may be more nanoparticle accumulation in the tumor cells, so there's more cisplatin release in those particular cells. That means that there's more DNA damage, more reductive oxygen species in that particular cancer cells apart from the other cells. So less systematic cytotoxicity as a whole, I would
Okay.
say.
Yeah, I would agree with that assessment. Andrea, so give us some insight, because I think that this was a little bit rushed there. Some insight about the nanoparticles and the ones that, or at least the compositions that seem to be more effective, and why do you think that's the case?
So the nanoparticles, had the most anti-cancer activity, but with good stability, was the C2, C8, C12 that had the more bulky ligand. I would wager that is likely tied to this being essentially larger, it being able, not larger, but like the fact that it's bulky is a strategy that's being used to try and reduce the toxicity in undesired locations in the body.
Okay, because that you not you, but Jad showed that the general metal uptake, which he found to be confusing, seemed to be pretty much comparable, right? So there has to be something going on for there to be more of an effect intracellularly. That's something that I would think. I want you guys to think about a little bit more. What structure? What is affecting in terms of reactivity, reaching the metal, reaching its potential targets? But yeah,
Can I
yeah, go ahead.
chip in?
Go ahead.
So it has to do with the C4 and the C6 are in secondary structures. They form secondary structures which are in the wires in the sheets, and they allude that there's more drug capacity inside of them. So there's a higher amount of cisplatin that is being held into these particular structures. So I would guess that it's a combination of, there's, even though there's comparable cisplatin concentrations, I mean, when you look at it statistically, there's comparable amount of platinum in the actual cytoplasm, but the secondary structure also offers a lot of more cytotoxic effects compared to just the C2, C8, etc., which are just small spherical particles. Because these secondary structures can inhibit other mechanisms of beta-actin or other proteins, which are critical for overall cellular health. So I think my main hypothesis would be that the C4 and the C6, that structural transform affects it really significantly, because we even see this. They have, I don't know if you want me to show you, but they also show the IC50 values of the different nanoparticles, and for the C4 and the C6, these are way higher. I mean, sometimes even 100-fold more active than even cisplatin.
Right,
So this
that's
has
what
to.
I was alluding to. That there has to be more going on. So you're then saying that these nanoparticles or that particular one is playing more of an active role in the effect on the cells, right? So yeah, that's essentially what I was going to. I will say, and this was from Keisha, there was a misconception. IC50 does not measure cell death. Okay.
Yeah.
It could indirectly give you some idea about it, but IC50 is the inhibition of the proliferation of cells. Okay, so it is not a measure of cell death directly. So I just want to correct that because that's a misconception I see a lot. All right, guys. So thank you very much. These were really nice presentations. I will send you a few additional questions from your classmates. So if you guys can respond to them. All right, then. Thank you so much. Then I'll see everyone next time.
Thank you.